Okay. Everybody ready to start with electrical activity of the heart? I understand what those graphs and TV shows mean. I have those green lines. Okay, so now that we have a little bit of an idea of what the anatomy of the heart looks like, we're going to be diving deeper into its function. So those concepts will be coming back. I'm going to start out by talking about the heartbeat. So we're going to be talking about how cells that create these electrical activities in the heart spread throughout the different parts of the heart and create contractions in the cardiac muscle. So to understand that, first we're going to talk about the cells that are doing this. So we say that the heart has this property called autorhythmicity. What this means is that the heart can generate its own rhythm, that we don't need to get information from nerves for these heart cells to create pulsations to beat. So you could put some heart cells in a petri dish and they would beat, which is pretty cool. That doesn't mean there aren't nerves to the heart. We will see that there are nerves to the heart that adjust the rate of heartbeat, but this property of the cells themselves exists. That's kind of like a backup system. Okay. So we saw that a little bit. I mentioned pacemaker cells with smooth muscle. Definitely have pacemaker cells in cardiac muscle, and we're also going to have another type of cell called conduction fiber cells. So these two cells together, these two cell types, I should say, are going to provide the beat of the heart and then the pathway where this current travels to get to all the muscle fibers that are actually doing the contracting. So there are a couple differences between these two cell types. So they are both going to be responsible for creating carrying current, but really the current is coming from pacemaker cells and then mainly traveling through the conduction fibers. Although in case of emergency, some of the conduction fibers could create a pacemaker. So what's going on with the pacemaker cells is that they're spontaneously depolarizing. So that means they're not waiting for a nerve to send an action potential, creating depolarization like we saw at our synapses. These cells just have this automatic cycle where they have certain channels opening on this kind of time loop. Okay. So we're going to take a look at that as we dive into pacemaker cells. So the point of these pacemaker cells is to coordinate and provide this rhythm to the heartbeat. The conduction fibers, their goal is just to spread this throughout the heart. And we're going to see that we have our current travel in a very specific order. So in order to make sure that blood has that unidirectional flow that we talked about, going through each of those different chambers in a specific order, that means we need to get this impulse to those regions of the heart in a specific order. Otherwise, we don't have this consistent flow of blood through these different locations. So we have some names for specific pacemaker cells and conduction fibers. And we'll go through it in a picture form, but basically we're going to start with the Sinoatrial Node. So that's going to be in your right atrium. This is the one that really sets the pace for the heart under normal function. It has the fastest beat, so the fastest one wins, kind of like you're jumping on a trampoline with a friend, right? If they're jumping faster than you, the trampoline vibrates even if you're jumping slower. Okay. So we're going to have our Sinoatrial Node. Then we're going to go through some conduction fibers to get to our next node. So we're going to go through our internodal pathways to get to our Atrioventricular Node. Internodal because it's going between two nodes. Then we're going to travel into the bundle of His, which we'll see is located approximately between our two ventricles, right and left. And then we're going to have conduction fibers that loop around and go back up the heart And this is so we can squeeze those ventricles from the bottom up, because we're trying to squeeze the ventricles up towards the arteries going away from the heart. So if we were to squeeze just from the top, you kind of get blood pooling in the bottom of the heart, and it wouldn't have any flow. So here are the firing rates for our autorhythmic cells. So as I mentioned, the Sinoatrial Node, or SA node for short, fires fastest. So it's firing at a rate of approximately 70 to 80 action potentials per minute. You'll see some variation there in different sources, but definitely fastest. Next, our sort of first backup plan is our atrioventricular node. It's firing at a rate of 40 to 60 action potentials per minute. So if your sinoatrial node stops firing for some reason, your heartbeat would slow, but it would keep beating because of this AV node. And then we have two more backup plans. So our conduction fibers could take over the heartbeat, but they're even slower. So that's what's going on with the bundle of His and the Pacemaker fibers. These different cell types are controlling the conduction. So this basically this action potential, right, within the structure of the heart. But we also need to spread those action potentials into the muscle cells that are actually doing the contraction. So like with our smooth muscle, we're going to see that cardiac cells have a lot of gap junctions. So they're going to pass the signal rapidly from cell to cell. And this is going to allow the heart to have this really synchronized contraction. We want all of those muscle fibers to be contracting at once. We don't want little tiny pieces of the atrium contracting independently of other parts of the atrium, for example. The rules are that the node or the fibers that are depolarizing fastest are controlling the other cells, because it's like a push, right, to everything. So we consider the fastest node, the SA node, the pacemaker for our heart. So let's take a look. We have someone talking about this.
So now we're going to discuss the cardiac conduction system and its components. This is a deeper dive into the biomechanical and electromechanical actions of this incredible organ called the heart. So now let's take a closer look at the heart and how it functions as a circulatory muscle. You see, the myocardium is the muscle that's unlike any other muscle within the human body. It's unique in that it can generate its own electrical impulse, known as automaticity. A special part of the heart, located in the superior aspect of the right atrium, called the Sinoatrial Node, or the SA node for short, works like an internal dynamic pacemaker. This internal pacemaker, when the heart is working correctly, generates an electrical impulse that then travels through the myocardium in a very organized and deliberate fashion. You see, this SA node generates an electrical impulse at about a rate of 60 to 100 times per minute. Now let's follow that pathway of the electrical impulse from the SA node to where it terminates at the end of the Purkinje fibers. So after the SA node initiates that electrical impulse, that impulse travels via the pathways called the internodal pathways. These travel throughout the right and the left atria, and then depolarizes the myocardial cells, which causes the actual muscle in the atrium to contract. From the atria, the electrical impulse travels along the pathway to the atrioventricular or AV node, where it may be delayed strategically before it moves through the bundle of His and ultimately to the Purkinje fibers, which travel down through and wraps around the ventricles, completing the electromechanical cycle of a complete heartbeat. See, the delaying the AV node, which is located in the left lower wall of the right atrium, is a necessary process in order to allow the ventricles to beat independently of each other, and thereby they operate as a double pump action. If for whatever reason the SA node does not operate properly as the primary impulse generator or pacemaker, the AV node then can begin sending its own electrical impulse instead. Though the AV node can generate its own impulse, it does so at a slower pace, which ranges between 40 and 60 impulses per minute. The impulse generated from the AV node then travels through the bundle of His, which is the route that the impulse travels to reach the Purkinje fibers, which wrap around those ventricles we talked about earlier. This ventricle contraction then circulates the majority of the oxygenated blood throughout the body. The bundle of His is the route of electrical transmission, which travels between the atria and the ventricles. Now, after the impulse reaches the bundle of His, it travels down the length of the interventricular septum and leads to the left and right bundle branches, of which the left bundle branch has two fascicles because the left ventricle is larger than the right ventricle. These bundle branches terminate into the Purkinje fibers, which depolarize the ventricular cells and cause the ventricular muscles to contract. In the case that both the SA and the AV nodes do not generate electrical impulses properly, the Purkinje fibers, which are located within the ventricles, then become the primary pacemaker source, which only generates electrical impulses in the range of around 15 to 40 beats per minute. Usually, this is too slow to produce an adequate systolic blood pressure or oxygenate cells within the body.
So part of why I keep showing you guys these videos is that as you'll see, as we add on to this information about the heart, we have a lot of things that are going on at once. And just looking at those two-dimensional pictures of the heart, I feel doesn't give you a really good idea of how the blood flow and the electricity are coordinated. So I recommend watching those videos again, but if they don't help, don't worry about it. Okay, so we're thinking about the excitation spreading through the cells, and our goal is to have the atria contract first, because that's where our blood is pooling, coming to the heart from our veins. And then we want our ventricles to contract, pumping blood through the arteries. Okay. So the coordination here is because we have gap junctions between our cells, which just allow these waves of electricity basically to spread from cell to cell, cytoplasm to cytoplasm. And we have these conduction pathways guiding us through the different parts of the heart. The feature of cardiac muscle that looks a little different that you wouldn't see in other types of muscle is something called an intercollegiate disc. So these are going to be the joining points between our myocardial cells, and they're going to have some specific properties that allow us to have this synchronized activity of the heart, as well as being able to resist the strong forces that we're going to be producing as we contract the heart to pump blood out. It takes a lot of force to get blood circulating through your whole body. Okay. So our cells join up kind of like this. Okay, so here we see the end of one of our myocardial cells and the beginning of the next one. So we can see that they look kind of like our other muscle cells, especially our skeletal muscle, right? So we can see that we have a sarcomere here, just like we did in our skeletal muscle, okay? But the intercollegiate disc is this space between our two cells, okay? The space between our two cells. So we can see that there are two special structures here in the intercollegiate disc. First one is our gap junction. So we can see how we have that conduction system spreading current through the gap junction. We could travel, have that yellow line travel all the way through a red arrow. We just have that conduction going from cell to cell rapidly through a gap junction, because travel through a gap junction is really fast because we don't have to worry about opening and closing channels. We don't have to worry about anything coming to a receptor. It's just direct cytoplasm to cytoplasm. And then the part that gives this muscle the strength to kind of resist these forces is something called a desmosome. So I suspect it's been a while since we talked about cell junction, but a desmosome is basically just a type of junction that physically holds cells together. Right, so this is for structural support. They just have bigger categories of this type of adherence junction. So that's what we see happening here. It's holding those cells together really tightly, giving them kind of like a skeleton almost. Okay. So this is different than a gap junction because we're not allowing things to go through. It's different than tight junctions, which we see in capillaries holding cells closely together, forming a seal. This one is giving us strength, basically. So if we're following the conduction system of the heart, we're following this electrical activity, we're going to start with our action potential in the SA node, in that sinoatrial node in the right atrium. This is where our blood starts, right? That's where blood, venous blood, is coming, collecting from the rest of the body. So our action potential is generated by these pacemaker cells in the SA node, the fastest of our pacemaker cells. And they're going to be spreading signal through the interatrial pathways into the atrial muscle and to the AV node. Okay. So I'm going to quick draw us a heart. All right. Not to scale. I know they never look like real hearts when you see pictures, but it sure is nice for drawing. Okay, so our SA node is somewhere like orange, it'll be brighter. Okay, our SA node in our right atrium, one of the things it's doing is going through the internodal pathway to our AV node, our atrioventricular node. But the other thing it's doing while it's doing that is we want the atria to contract. They contract before the ventricles to contract. That's how we get blood into the ventricles in order to squeeze it out through our arteries. So it kind of has like, you can imagine them as like off ramps, basically. So we'd have other fibers allowing the atria to contract. And when we have these contractions, and that might not be immediately obvious based on the flow of blood that we talked about, that both atria are going to contract basically at once, and then both ventricles are contracting at once. So we're like squeezing the top of the heart and then squeezing the bottom of the heart in order. So two of these compartments together. Okay. So once we are at the AV node, so we went from the sinoatrial node to our atrioventricular node. We're traveling through the bundle of His, which does split. So it kind of has a bundle branch to the right and to the left, going to each ventricle. That's why they're called the atrioventricular bundle. And then the left and right bundle branches. Right. And then we have our Purkinje fibers looping up around so that we squeeze from bottom up. So it's really those Purkinje fibers that are going to be giving the signal to most of the ventricular muscle. So we can look at this in diagram form as well. So here we see the sinoatrial node up at the top here, right, top of the right atrium, right kind of in the attachment point for our vena cava, bringing our venous blood to the right atrium. Going through our internodal pathway to our AV node, right here, kind of the separation between the atria and the ventricle. Next, we have our bundle of His, so our also known as our atrioventricular or AV bundle. Some of these things have a couple of equivalent names. You may have noticed in the video we talked about automaticity instead of autorhythmicity. Those are the same thing. So AV bundle and bundle of His are equivalent terms. Okay. But then splits into bundle branches. I don't think I mentioned the name of this structure before when we were looking at heart structure, but what is traveling through there, that wall, is the interventricular septum. So the septum, like the septum of your nose, is a wall. So it's the septum between the two ventricles. Okay. So we're traveling through there. And then our Purkinje fibers are looping us up and around. It should not be surprising that ions have something to do with this process, right? So just like when we talked about neurons, we had ions controlling our action potential in our neurons. We have ions traveling in and out of these pacemaker cells as we create these depolarizations. So even though they're spontaneous, we still have ions traveling. We're still thinking about that membrane potential, that difference in charge across the plasma membrane. The only difference here, well, there are a couple of differences, but the first difference is that these are spontaneous. And then the second is that the action potentials here are going to look different, and they're going to a little bit different in graph form, and then also the specific ions involved. We're going to talk about this in a little less detail than we did with neurons. Your book goes into some more detail on additional types of channels, like funny channels. We're not going to worry about those for our purposes. We're just going to focus on the named ions that are hopefully familiar to us, although they're doing a little bit of a different thing here. Okay. So the spontaneous depolarizations that we see in our pacemaker cells are going to be caused by closing potassium channels. So that part is kind of familiar from our neurons. We're going to be opening channels as well. We're going to be opening sodium channels like before, but here calcium channels are going to be really important. So we're opening sodium channels, but we're also opening calcium channels. All of this is going to have the effect of depolarizing these cells, just like in our neurons, depolarization, that is an action potential, right? A big wave of depolarization. So here, the part that's really the fast part of our action potential is going to be reliant on calcium. So in our neurons, we had a lot of sodium flowing in. This time we're going to have a lot of calcium flowing in. Our repolarization is like our repolarization phase of the neuron. It's those potassium channels open. Okay. So the colors are kind of hard to see on this slide, so I'm going to trace us through, but so if we're looking at the action potential here, it looks in shape mostly like our other action potentials, right? So we have our first depolarization. This is happening slowly, spontaneously. Then we have a rapid depolarization, creating a peak of our action potential. Then we repolarize. We repolarize all the way, and then we come back up to threshold. So the part that's different here is what is changing in terms of ionic permeability. So what we have happening here at first, at the beginning of our slow depolarization, before we get up to thresholds, we have the permeability of potassium decreasing. Potassium is not leaving the cell as much, and the permeability of sodium is increasing, so we have sodium coming in. Okay. So note that even though sodium is coming in a neuron, that's what caused our spike, but here in our pacemaker cell, that's not causing a spike yet. Then during the second part of our action potential here in our pacemaker cell, our permeability to sodium is actually decreasing, so it's dropping. Basically, sodium isn't in control of this depolarization anymore. It's the calcium. So the permeability of calcium is increasing here. And that is what triggers our spike. So we still have to get to a threshold. And once we hit that threshold, we have a lot of calcium flow in. Right, so that's our rapid depolarization, is the calcium. And then we turn the system off at the end, right? So we start to decrease the permeability of calcium and increase the permeability of potassium. overall process, the differences here are kind of on the upswing, basically. Questions about this process? Okay, so I want you to note specifically that this is happening in a pacemaker cell, because as we're talking about the heart, we have our different cell types, so different things are happening in different cells. So this is our pacemaker cells specifically. I say that because now we have to talk about the contractile cells. So these are the ones that we think of with the sarcomeres, actively contracting, making these different chambers of our heart smaller. So we're going to have 5 phases of electrical activity in the contractile cells that are going to look a bit different than what's going on in those pacemaker cells. And the pacemaker cells are just sending the signal out, but then when the contractile cells get the signal, they have to create their own action potential, and that just looks a little different. So we call them phases 0 through 4. So you've never seen a list starting with 0, now you have. Okay. So first we're going to increase the permeability to sodium. Next, we're going to turn off our sodium and increase the permeability to calcium instead. At this point, we start decreasing the permeability to potassium, so it's escaping the cell more slowly. And then we're going to increase the permeability to potassium, letting it leak out, decrease the permeability to calcium, not coming in so fast, and then we get back to rest. And we'll look at this on the graph form as well. A thing to note about the contractile cell action potential is that it's much longer in duration than skeletal muscle. So that's also part of what's creating these heartbeats, right? This pace of contraction. Right? So here we see our action potential in the contractile cell looking kind of different, right? So we don't have this slow depolarization to threshold happening. We start pretty immediately with our big spike, our big depolarization. So this big depolarization, this number 0, got to remind myself as well, right? So the first thing that happens is that increased permeability to sodium. So now we're kind of, in terms of ions, that's similar to what we saw in neurons, right? So our big depolarization in neurons and in these contractile heart cells, the responsibility of sodium compared to the pacemaker cells where it was really the calcium. Okay. So then we start to have this slope down, and this part looks a little different than we've seen before as well. So we start to have a brief repolarization first in this phase 1. So what's happening here is that those sodium channels are closing, right? So we start to repolarize, but then our calcium decides, never mind, I do need to play a role in this, right? So here in phase 2, we have increased permeability to calcium. So basically, even though we're not letting sodium flow into the cell anymore, some calcium starts coming in, which is why we don't continue dropping down, right, in the same way, right? So that's what would happen if we didn't have calcium coming in, but instead we're following this other line. Right, so instead of going straight down, we start to plateau because that influx of calcium keeps our membrane potential up for a little while. But then as the permeability to calcium decreases and we allow potassium to flow out of the cell, we finally actually repolarize down to our resting level. So this is specifically happening in those contractile cells. So this is what the sort of stereotypical muscle fibers are doing in our heart, rather than just those kind of highways of electricity. So as we send this action potential through our heart cells, the contractile cells, we're also going to have excitation contraction coupling. So like in our skeletal muscle, right, we have an action potential come to the cell and it's traveling through the cell to neighboring cells, but it's also causing that cell to contract. So those properties are going to be similar to skeletal muscle. As we run through that, it's going to look a lot the same. We're still going to have T-tubules. We're still going to have a sarcoplasmic reticulum that releases calcium. And we're still going to have troponin and tropomyosin regulating basically our cross-bridge cycles. But we're going to have some components of smooth muscle here as well. Mainly, we see those gap junctions. We didn't see that in skeletal muscle. And some of the calcium that we use for this process is going to be coming from outside the cell, not all coming from the sarcoplasmic reticulum. Okay, so calcium didn't really come from outside the cell much in the skeletal muscle, but in cardiac and smooth muscle it does. We're going to run through this diagram. So we're going to start out as we get this signal. So we're saying that this specific contractile cell that we're looking at is getting this signal from a neighboring contractile cell. Right, so this is a current that's spreading through the heart muscle, through those contractile cells themselves. Right, so it's just getting that depolarizing current, that action potential coming through its neighbor. So first we're going through a gap junction into our cell of interest. Okay. And then we would have that whole complex steps 0 through 4 happening as we have this action potential traveling to the next cell. But at the same time, it's going to be causing that cell to contract. Okay, so our second step is that our action potential is traveling. And as it's traveling, it is going to hit a T-tubule, which kicks off our contraction, basically. So it would continue traveling and give this signal to the next cell, but it also causes contraction. Okay. So as it's causing this contraction, it's traveling down the T-tubule. And as it does that, we have a bunch of calcium channels opening. So the calcium channels are opening in 2 places. The first place they open is here on the plasma membrane. They're also opening in the sarcoplasmic reticulum. So we're getting calcium ions from both of these sources into the cell, allowing it to contract. So now we've got calcium in the cell. This means, like in our skeletal muscle, we have that calcium bind onto troponin, which exposes the myosin binding sites on the actin. Right, so we can see that's just our normal looking actin that we talked about with skeletal muscle here too. And then we have our cross-bridge cycle happening, just like it did in skeletal muscle. And finally, the calcium, some of it gets transported back into the sarcoplasmic reticulum, and some of it actually gets pumped actively out of the cell. So we're taking some of the calcium out into the extracellular fluid. Because we have to regulate these calcium levels because this is also part of how we're sending that action potential to the next cell. So we gotta keep it under control. The questions about this process. Hopefully kind of familiar since most of these topics are just slight changes to things we've seen already. Great, and our tropomyosin is blocking this back and the muscle fiber is relaxing. So the relaxation of those contractile cells, those heart muscle cells, are happening as we remove the calcium, right? Because the calcium is necessary for that cross-bridge cycle, right? So in skeletal muscle, that was how we relax. Same thing happening here in the heart. The only difference is that we are both sucking that calcium back up into the sarcoplasmic reticulum and sending some of it into the extracellular fluid. So some of that is happening through a calcium ATPase, right? So it means we're directly using ATP to pump that calcium out, right? Our primary active transport. And then we also have secondary active transport happening. We're using a sodium gradient in order to exchange calcium. the troponin and the tropomyosin are doing their normal jobs related to the axis. All right. So a couple of review questions on what we just talked about. Right. Make good guesses. Or answers. Great. Yeah, we have our pacemaker cells and our conduction fibers creating this rhythm. So where are those pacemaker cells? Seems like we're all on the same page. So that's great. Hopefully this one's easy too. Okay. Awesome. Yeah, our atrioventricular and sinoatrial or AV and SA nodes are where we have our pacemaker cells. Usually the SA node is the fastest, so it's the big pacemaker for the heart. But the atrioventricular node can take over if we need it to. Okay. So finally, we're just going to talk about how we can actually get information about the heart and the function of this electrical activity. As I think you'll see in lab, I think we have someone with a pacemaker in our donor body lab, right? Sometimes things go wrong with this system. Sometimes these pacemaker cells don't work the way we need them to. So sometimes we actually have to make interventions and put actual, mechanical components to kickstart these fibers. So as we're testing whether the system is working right, we're going to put electrodes on people's bodies, on the skin, in order to measure the heart. Now we can do that because your body is mostly water, right? So this electrical activity, even though it's not sending off action potentials willy-nilly in your body, you can measure it with an electrode. So just like you've been doing in phys lab, right? You can measure where electricity is happening. So you'd have a positive end and a negative end, right? Like kind of a ground, and you'll be measuring the difference between two places on the body. This is non-invasive, which is great. And we can test for clinical abnormalities using this non-invasive technique. Okay. So we call this thing an electrocardiogram. This is an external measure of the electrical activity of the heart. Right? Electrical, cardio, heart, gram, like a graph. Our body is the conductor. We use the body as a conductor in a number of different types of graphs, right? So this is an ECG, but we also do this in our EMGs and our EEGs, which measure the electrical activity of muscles or the electrical activity going on in the brain. And so using the body as a conductor is a useful process for a lot of different conditions. So the distance and amplitude of the spread, basically properties of this graph that we're going to see, going to be dependent on the size of the potentials, right? So how big these action potentials are, and then also how synchronized these potentials are between our cells. Now, looking at the heart is pretty easy on that second point. The heart is really synchronized. So that's really helpful for getting a clear diagram, right? A clear graph. So we're going to see something that looks kind of like this. So this first image should be kind of familiar. What type of cell are we measuring there? Membrane potential, what's it looking at? Yeah, the actual contraction of those heart cells. So that's the membrane potential of the contractile cells. So that is what you would measure if you were trying to measure the actual muscle, right? But what we're measuring with our electrocardiogram is going to look like this. So this is what we're going to see from basically the outside of the body. This is what we're going to get from our various electrodes that we place. So it has this very stereotypical shape. So we're going to be looking for specific waves happening in this order. So first, we see here our P wave, right? So first we get this P, right? This peak. Next, we have a Q, so our Q segment here at the bottom, basically. Our R is up at the top, and S, going back down, and then we wait a little bit, and we should see a T. So each of these different peaks and valleys is going to correspond to a specific kind of action in the heart. So it's going to be linked to electrical activity of the heart, but that electrical activity of the heart, because it spreads out through the muscle cells, right, should also tell us something about which parts of the heart are contracting at what times, right? Okay. So it's our PQRST complex. We're going to look at different parts of it to figure out what's going on in different sections of the heart. So the first thing that happens in your heart is that you have the atria contract. So that P wave, the first one, what that is looking at is depolarization of the atria. Right? So the first thing that happens is this little spike here in the atria, that first part of the heart. So you can see that that is not the tallest peak here, but it's super important. We need those atria to depolarize first because we need that blood to travel in the correct order. Next, we often refer to the QRS complex as a whole. So that's this valley, then this peak, and then going back down. Okay. So what we're seeing here in this QRS complex, the reason we have this big spike is basically because of our ventricles depolarizing and contracting. But at the same time, we have our atria repolarizing, right? So those little valleys are also because the atria are going down, right? They're beginning to repolarize. Okay? So during the QRS phase, we should see the atria kind of relaxing and the ventricles contracting. And then during our T wave, we have the ventricles repolarizing. Okay. And so we can look at different components, so spaces between these, to think about more detailed things as well. So one thing we might look at is the difference between P and Q. So the difference between P and Q is going to tell us something about how long it's taking that conduction to get from our atria, right, like from our SA node to our ventricles. So how long it's taking us to travel to that AV node and how long the AV node is kind of pausing before it tells the ventricles to contract. Other things we might look at, we could look at the distance between Q and T. So what overall would happen between Q and T, do you think? And it says ventricular systole, but what does that mean? What's happening between Q and T?
Ventricle contract.
Yep, Yeah, so the ventricle is contracting, pumping the blood, and relaxing. That's the T part. So that's like the whole cycle of what's going on in a ventricle, is that squeezing and that relaxation. Right? So we're going to refer to that as ventricular systole, that whole time that it's basically contracted or partially contracted. Okay. And so then our other thing we would want to measure is the difference between T, so the end of that contraction basically, and our next segment. So between T and, I should make that line actually a little longer. Right. So between T and the next Q, so the next kind of beginning of our QRS segment. Okay. So that's the time when the ventricles are going to be basically fully relaxed and chilling, right? They're just hanging out. So when you see people refer to systole and diastole, like in a clinic and you're measuring these things, really we are talking about ventricular systole and ventricular diastole, because that's the big one, right? That's the big one, and it's what's pumping the blood out to your body. Great. So if we have a last question, and then we are finished with this stuff. Really love the heart. I hope this is all coming across clearly, but if it's not, always welcome to ask questions. I know the first time I learned the heart, I was super confused. Now it's hard to remember that. But that's really cool. Okay. Final answers. Let me click through. Great. Yeah, our ventricles are depolarizing and our atria are repolarizing. So next time we're going to start pulling this all together, and we're going to be talking about the cardiac cycle, and we're going to move into cardiac output.